Hi class. So today we're going to go over the cell cycle notes. We're going to be talking about cell growth, cell division, and cell regulation. So where it all began. You all started as a cell smaller than we see at the period of this sentence. And then over time you grew and developed. So that process of growing and developing is known as cell division in terms of your cells growing and developing. It's the continuity of life, meaning that life depends on cell reproduction. Cells must divide for reproduction, such as this single-celled amoeba who is making a copy of itself, for growth, for repair, and renew. The cell cycle is the life of a cell from origin to division into two new daughter cells. So from one cell to the next. From one cell, we get two new cells. What is passed on to the daughter cells? Well, the exact copy of genetic material is known as the DNA. DNA must be transferred from one cell to the next. That step is known as mitosis. The assortment of organelles in cytoplasm must also be duplicated. That division is known as cytokinesis, or division of the cytoplasm. When we are copying the DNA, we divide the DNA into two. So the dividing cell duplicates the DNA. It separates each copy to opposite ends of the cell. In essence, during the copying process, we split the DNA into two daughter cells. As a side note, human cell duplicates around 3 meters of DNA, which is about 2 football fields. We separate two copies so that each daughter cell has complete identical copies. The error rate of copying DNA is less than 1 per 100 million bases, which is actually really good because there's about 3 billion base pairs in our genome. So that gives us less than 30 errors per cell cycle. A little bit about DNA, which you're required to know, is that DNA is organized in chromosomes. So if I were to untangle a strand of of a chromosome, it is a long strand of DNA. DNA wrapped around histones is known as chromatin. And these histones help to condense the DNA, or it helps to shrink it down. After DNA replication occurs, the chromatin starts to condense. Basically, the coiling and the folding to make it into a smaller package. So from DNA to chromatin to highly condensed mitotic chromosome, as we see here. You also need to know the structure of a chromosome. So a chromosome is a duplicated chromosome consists of what is known as two sister chromatids. You get one chromatid from each parent, so one from your mother, one from your father. It is held together at the centromere, or in the middle, and it contains identical copies of the chromosome's DNA. The cell cycle has a life cycle. So when your cells are formed, the cells have an option of to do one of two things. The cell's either going to grow and mature and divide again, or the cell's going to grow, mature, and then never divide again. Okay. If the cell is a type of cell that grows, matures, and divides, it's going to go through these stages here, the G1, S, G2, and the mitosis phase. Those types of cells are going to be our somatic cells or our body cells, such as epithelial cells, blood cells, and our stem cells. However, if it is a cell that's going to never divide again, these cells are only working, and they are placed in what is known as the G0 stage. Okay? That would include our brain and our nerve cells. These cells never ever divide. So they're stuck in this resting phase, but they're not really resting. They're just working. They just never get called into division. Okay. However, there are some cells which are exceptions to the rules, and that would be our liver cells. Our liver cells are arrested in G0, meaning that they're in a working, non-dividing stage. However, if they sustain some sort of damage, they can be called to regenerate themselves. Okay, they can be called back. Here's a video on how a wound heals itself. So I would suggest that you watch this, and you could see basically how our cells divide to heal a wound. All right, the phases of a dividing cell's life includes interphase and the mitotic phase. Okay. During the interphase, the cell grows It replicates the chromosomes and it produces new organelles and biomolecules such as carbohydrates, proteins, lipids. Okay. Once it finishes interphase, then the cell is going to enter mitosis, as we see here. During the mitotic phase, the cell separates and divides the chromosomes. The nucleus divides during the process of mitosis, and then the cytoplasm and organelles divide during the process of cytokinesis. Here's our control of our cell cycle diagram. Please make sure to read through each of these stages, our GAP1 synthesis GAP2 phase, as well as G0 phase. These all make up for interphase. As we'll see in the next slide. Here's a video on our amoeba sisters. Same thing, please review this on your own. This is a really good video to help you guys understand the mnemonics for mitosis. All right, let's start with the first phase, interphase. 90% of your cells are in interphase. So the majority of your cells are actually just working. They're doing their everyday job. They produce RNA, they're synthesizing proteins, and then they prepare for duplication if it's triggered. Okay. The characteristics for cells that are in interphase is that they have a nucleus that is well defined, and the DNA is loosely packed in long chromatin fibers. So like we said, the majority of your cells are going to be in this stage. Interphase is divided into 3 phases: GAP1 or G1, synthesis, and GAP2. So during the GAP1 or G1 phase, the cell is working, it's doing its everyday job, and it's growing. During the S phase or synthesis phase, the DNA is copied. And then finally, during the G2 phase, the cell prepares for division. It grows and it produces the organelles, proteins, and membranes. Once it completes all of these stages in interphase, then it can be triggered to start mitosis. Mitosis is the copying of the cell's DNA and dividing it between the 2 daughter nuclei. Mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase, or as I like to say, PMAT, P-M-A-T, prophase, metaphase, anaphase, and telophase. Here's an overview of our cells going through mitosis. So as we said, they start in interphase, prophase, metaphase, anaphase, and telophase. Let's go ahead and look at each one individually. During the process of prophase, the chromatin starts to condense. Centrioles, which we see only during cell division in animal cells, move to opposite poles of the cells. And what centrioles do is they attach these fibers across the cell to form the mitotic spindle. The nucleolus then starts to disappear, and the nuclear membrane starts to break down. These proteins then are attached to the kinetochores, which are focused or which are located here on our chromosomes. So these fibers will attach here, and the purpose is to move the chromosomes around. Remember, the process of mitosis is to split the cells in half. So everything has been copied already. Okay. During metaphase, the spindle fibers align along the chromosomes, along the middle of the cell. So as we could see here, the chromosomes line up in the middle. Remember, they have been duplicated. So the purpose of this stage is to start to separate the duplicated chromosomes. Okay, they line up in the metaphase plate, and these help to ensure that the chromosomes separate properly. So the reason why they line up in the middle is to make sure that each set of cells is going to be receiving the same amount of DNA. As we see here, chromosomes line up in the middle. During anaphase, the sister chromatids start to separate. They move to opposite poles, and they are pulled at the centromeres. They're pulled by motor proteins that are walking along these microtubules. Now, during anaphase, we see that there's an increased production of ATP by the mitochondria. Well, why is that? Because these molecules here are now starting to move. Anytime we have movement, we need ATP because movement generates energy. Finally, during telophase, the chromosomes arrive at opposite poles, the daughter nuclei form, and the chromosomes start to disperse. The spindle fibers as well start to disperse, or they start to disappear, and then finally cytokinesis starts to begin. Now, as we see, these cells are still connected. So the cell's still connected. It has not completely been separated as of yet. During cytokinesis in animal cells, a cleavage furrow forms, and this is when the cell's starting to separate. Remember, it's separating the cytoplasm. Here's a picture of the cells in cytokinesis. As we see here, the cleavage furrow starts to form. Here are cells of mitosis in a whitefish blastula. Notice interphase, we see the nucleus. Prophase, the chromosomes appear. Metaphase, they line up in the middle. Anaphase, they start to separate. This one isn't a very great picture of anaphase. It looks more like metaphase. Telophase. Here's our same pictures. All right, cytokinesis in plants, how is that different? Okay, the vesicles move to the equator, they line up and they fuse to form two membranes, which is known as the cell plate. That is then derived from the Golgi. The cell plate will eventually form what is known as the cell wall in the plant. It will form the cell wall in the plant. So here we go with cytokinesis with our plant cells, cytokinesis with our plant cells. Same thing, plants go through interphase, prophase, metaphase, anaphase, and telophase. Same thing. All right, the evolution of mitosis. Mitosis in eukaryotes likely evolved from binary fission in bacteria. Okay? Binary fission is the process in which a bacterial cell makes a copy of itself. And during that process, the single circular chromosome makes a copy of itself. There are no membrane-bound organelles. Once the DNA makes a copy of itself, as we could see here, replication begins and the cell divides into two. And the cell starts to divide into two. Here's a video on binary fission. Hopefully this will help you have a little bit more better understanding of binary fission in bacteria cells or bacterial replication. All right, coordination of cell division. Multicellular organisms need to coordinate across different parts of the organism. So some of the things they coordinate is going to be the timing of the cell division and the rate. So timing and then the rate. This is crucial for normal growth, development, and maintenance. So do all cells have the same cycle? And that would be no. Depending on the type of cell that we have and its function is going to determine whether it is going to divide or not. The cell cycle is controlled by internal and external signals. So a signal is a molecule that either stimulates or inhibits a metabolic event. Some internal signals would be family of proteins which are called cyclins. And if I look at the term cyclin, cyclin has the word cycle. And they increase and decrease as the cell cycle continues. Without cyclins, the cell cycle would stop at either the G1, mitosis, or G2 checkpoints and allows times for any damage to be repaired. Apoptosis is a term that you need to know, so make sure that you highlight that. Apoptosis means programmed cell death. And some of our cells are programmed to die, so that would be a slowing or stopping of cellular division, right? It involves a sequence of cellular events. Either the nucleus gets fragmented and breaks apart, the plasma membrane or cell membrane blisters, or it engulfs cell fragments. Apoptosis is caused by enzymes which are called capases. Mitosis and apoptosis are opposing forces, meaning that mitosis increases cell number, whereas apoptosis decreases cell number. Here's some illustrations of apoptosis as we see here. Apoptosis harbors capases, like we said, which are the enzymes that are kept in check by inhibitors. They can be unleashed by internal or external signals, meaning that these enzymes can either get a signal from the outside or inside environment. They signal protein P53. P53 is our guardian protein, our guardian enzyme. We need it. And what it does is it stops the cell cycle at G1 when DNA is damaged. So if your DNA becomes damaged in any way, shape, or form, then P53 will detect that and will stop the cell from dividing. It will initiate an attempt at DNA repair. So before the cell can continue to divide, it's going to try and repair the DNA. If it's successful, then the DNA will continue to mitosis. If it's not, then P53 will signal apoptosis, and the cell will basically die. So here's our regulation as we see at our G1 checkpoint. So notice that there's no DNA damage, breakdown of P53. Now if P53 detects that the DNA has been damaged, then it's going to go ahead, initiate it, and it's going to fix the DNA. It's going to bind to the DNA and it'll fix. Frequency of cell division varies with cell type. So skin cells divide frequently throughout your life. Liver cells, as we said, retain the ability to divide, but they keep it in the reserves. Mature nerve cells and muscle cells do not divide at all after maturation. Now, there's two irreversible points in the cell cycle, meaning that once these occur, we can't go back. One is replication of genetic material. So once DNA is copied, you can't undo that. And then two, separation of the sister chromatids. So once the sister chromatids separate, you can't move back as well. Cells can be put on hold, though, at specific checkpoints. These checkpoints control the cell system. So cell cycle controlled by a stop and go chemical signals at critical points. These signals indicate if there's key cellular processes have been completed correctly. So notice that we have a G1 checkpoint, we have a G2 checkpoint, and then we have a mitosis checkpoint. During G1, we ask, can the DNA synthesis begin? Is it ready for DNA to make a copy of itself? During the G2 checkpoint, has DNA synthesis been completed correctly? Meaning, has the DNA copied itself properly? Then is it ready to commit to mitosis? And then finally, during mitosis, the spindles are going to be checked to make sure that they have attached onto the chromosomes properly, and can the sister chromatids separate correctly? The G1 checkpoint is the most critical of all. It's the primary decision point as to whether or not the cell is going to divide. That's the restriction point. If the cell receives the go-ahead signal, then it's going to go ahead and continue on to division. If it does not receive a go signal, the cell will exit the cycle and it switched to the G0 phase, which is a non-dividing stage. So that cell will be signaled you don't have the necessary items needed for you to complete the cycle. So now we're just going to put you in this resting stage, Okay, the G0 phase is a non-dividing differentiated phase. Most human cells are in the G0 phase. So some cells that are in the G0 phase would be like our liver cells, right? So they're in G0, but they can be called back to the cell cycle by external cues. So if there's some sort of damage to the liver cells, they can then go back to the G0 phase. Nerve and muscle cells are highly specialized cells, so they are arrested in the G0 phase and they never divide. So how do cells know when to divide? This is through the process of cell communication or through signals. So there's a chemical signal in the cytoplasm gives the cue. These signals usually mean proteins, or the ligand. They're either an activator or an inhibitor. So it's either going to tell the cell to divide or it's going to tell it to stop dividing. The signals that to promote cell growth and division are our proteins, our internal signals, and our external signals. So we have promoting factors and growth factors. The primary mechanism of control is through phosphorylation through kinase enzymes. So what that means is once these enzymes receive a phosphate, it phosphorylates and then it sends the signal necessary for the cell to divide. The promoting factors are known as cyclins, CDKs, and APCs. The ones that you're going to be most familiar with that we will be looking at will be the cyclins. And whenever you see the term cyclin, I want you to think of cycle. Basically, these promote the cell cycle. CDKs are cyclin-dependent kinases, and these are enzymes that activate cellular proteins. They promote mitosis. APCs are anaphase promoting complexes. We won't focus too much on those, but like I said, we will be looking most at the cyclins and the CDKs. So once those are turned on, then that means that your cells signal to start dividing. These are the different types of checkpoints as we've seen here and the enzymes involved for signaling. So cyclin and CDKs are going to be submitted or released during the G1 checkpoint. CDKs and cyclins, or the MPFs, are going to be submitted during G2, and then the APC during our mitosis checkpoints. CDKs and cyclin drive the cell from one phase to the next in our cell cycle. So these need to be properly regulated so that the key to life, that these genes for these regulatory proteins have been highly conserved through evolution. So CDKs and cyclins, we see that these proteins have been conserved over time. So these are highly, highly important proteins. The genes are basically the same in yeast, insects, plants, and animals, including humans. So CDKs and cyclins, these proteins are the same as we see in yeast, insects, plants, and animals. Basically, these are responsible for turning the cell cycle on and promoting mitosis or cellular division. All right, what are some external cues? So growth factors, as we stated, these are an external cue. So proteins are signals released by body cells that stimulate other body cells to divide. I get a cut, great. Now I need to promote cellular division, okay? We have two types of cellular inhibitors that we look at. We look at density-dependent inhibition and anchorage-dependent inhibition. So crowded cells stop dividing. So in density-dependent inhibition, crowded cells stop dividing, meaning like your cells regularly know when to stop dividing. So if I get a cut, cells are going to multiply, cyclins and CDKs are going to be released from your cells to promote cell division. Your cells need to know when to stop dividing. So the mast cells use up what is known as growth factor. There's not enough left to trigger cell division any longer, and then your cells stop dividing. It's also known as anchorage dependence. So to divide, cells must be attached to a substrate. So they attach to each other. So if I were to see here, if I were to take out normal cells, notice here that they're going to grow, and they're going to continue to grow, and they know when to stop growing, okay? Here's our signal transduction response, as we've seen here. Growth factor is here. It's then going to promote these protein kinase cascade, which will then tell the cell what to do and initiate cell division. All right, here's a brief videos on cancer biology. It's going to talk about what happens when the signal is disrupted. When the signal is disrupted, I do want you guys to please pay attention to these videos as they are really well put together. An example of growth factor would be platelet-derived growth factor, or PDGFs. Okay, these are made by platelets. Platelets are located in our blood, and what platelets help to do, you guys, is they help to form blood clots. So bindings of PDGFs to cell receptors stimulates the fibroblast or connective tissue to cell development, which aids in wound repair, as we see here. So if I were to cut up a sample, as we see here, of connective tissue into small pieces, I then obtain suspension of free fibroblast cells by using enzymes to digest the extra cellular matrix. Then I transfer these cells to a sterile culture. These cells adhere to the glass and they start to incubate over time. Notice here that the cells that are in basic growth medium plus the PDGF, the cells start to grow. Without it, we can see that they stopped. So PDGF, like we said, helps cells in wound repair. All right, growth factors influence cell cycle. Everybody here is born with cancer genes. They are known as proto-oncogenes and tumor suppressor genes. The key is to figuring out what turns them on and turns them off. So normal genes that become oncogenes when they are mutated are known as proto-oncogenes. They stimulate cell growth. If they are switched on, they can cause cancer. So these genes in our bodies, obviously the majority of us, are turned off, okay? If the switch turns on, then it stimulates cell growth. An example is RAS. RAS is an example of a gene that helps to activate cyclin. Cyclin, like we said, turns on cell division. The other gene we all have is known as a tumor suppressor gene. And what this does is it inhibits or it stops cell division. If it's switched off, it could cause cancer. Our example here is P53. If P53 is not functioning like we said, it doesn't recognize damaged DNA. If damaged DNA is allowed to duplicate, then it can cause mutations, which can then cause cancer. Cancer is essentially a failure. It's a failure of cell division control. It's unrestrained, uncontrolled cellular growth. So what control is lost? Basically, all of the checkpoints are ignored. The G1, the G2, and the M checkpoint. P53, like we said, plays a key role in the G1 checkpoint. P53 protein halts cell division if it detects damaged DNA. It stimulates your repair enzymes, as we said, to fix DNA, and it's either going to force the cell into the G0 resting stage, or it's going to keep the cell in the G1 arrest stage, or it's going to cause the cell to kill itself through the process of apoptosis. All cancers need to shut down P53, so this enzyme is highly, highly, highly important. Here's an illustration of how P53 functions. Notice here, in our normal P53, our DNA is damaged by sunlight. P53 then is going to recognize the DNA damage. It's going to do one of two things. It's either going to fix the DNA, and then once it fixes the DNA, it's going to allow the cell to divide because the DNA has now been fixed and altered. If the DNA is damaged beyond repair, then it's going to cause the enzyme to trigger apoptosis, and then the cell will die. Okay. If P53 is shut down, the cell doesn't even recognize that it has mutated DNA. The cell is going to continue to divide, and over time, the cell can turn cancerous. Here's a cool video on the role of P53. Please, please, please watch it. It is really, really good. It actually shows you a nice computer image of how P53 works to fix the DNA. All right, development of cancer. If you did not listen to me at all, this is the one thing that you should know, okay? If there's anything you need to know, this is a slide that you guys need to know. Cancer develops only after a cell experiences at least these 6 key mutation hits. It has unlimited growth, so these cells grow out of control, meaning that the growth promoter genes are turned on. It ignores the checkpoints, so we turn off the tumor suppressor genes. It escapes apoptosis, so the suicide genes are turned off. These cells are immortal. They have unlimited division. So we turn on chromosome maintenance genes. These cells also promote blood vessel growth. So we turn on the blood vessel growth genes. And they overcome the anchor and density dependence. So we turn off the touch sensor genes. So these cells don't know when to stop dividing. Here's a video on what happens when DNA is damaged. Same thing. Please watch this video on your own. All right, what causes these hits? Mutations in cells can be triggered by UV radiation, chemical exposure, radiation exposure, heat, cigarette smoke, pollution, age, and genetics. So any one of these things can cause the cells to mutate. If they mutate, then these genes can be turned on. All right, tumors. There's 2 different types of tumors you should know about. Tumors are basically masses of abnormal cells. We have benign tumors and malignant tumors. In benign tumors, the abnormal cells remain at the original site as a lump. P53 has halted cell division in this instance. Most do not cause serious problems and can be removed by surgery. Benign are also known as what science community would call friendly tumors. Friendly tumors meaning that these aren't going to grow. They're not going to go anywhere outside of the body. They will stay contained. They don't travel. Whereas malignant tumors, the cell leaves the original site. It loses the attachment to nearby cells. These can then be carried by the blood and lymphatic system to other tissues, and they start more tumors or they metastasize. Eventually, they impair the function of the organ throughout the body. They disrupt the organ, and then eventually, if it cannot be killed off, then these obviously can overrun organs and stop them from functioning. Some traditional treatments for cancers. So treatments target rapidly dividing cells. We have high energy radiation and chemotherapy with toxic drugs. They kill rapidly dividing cells. So that's why when a person is taking chemo, your hair is a fast-growing cell. Chemotherapy, like we said, stops rapidly dividing cells. So people that have chemo, they lose their hair and their skin kind of looks like almost a little burned in instance. That's due to these toxic type of drugs, okay? Because we want to try and kill off all of our cells, all of the cells, in hopes that we kill off the cancer cells as well. Same thing with radiation. Radiation only targets a specific area, though. Radiation only targets one specific area. Chemotherapy targets all cells. There are also numerical drugs, though, that are in treatments. So these are drugs that target the protein or the enzymes that are found only in tumor cells. So if we could disrupt the cell from dividing, then we would slow the cell from replicating, which would then slow the spread of these cancers. There's a treatment which is known as Gleevec, and it's the first treatment for adult leukemia and stomach cancer. And it was the first successful drug that basically disrupted the cell communication. So as we can see, when this enzyme here is disrupted, it stops the production of the cancer protein, which will then stop cells from dividing. And those are the cells that we want to stop dividing, are these cancerous cells. Here's some more videos, TED Ed videos. These are pretty good. How does cancer spread? Why is it so hard to cure cancer? Chemo versus cancer, and then hacking bacteria to fight cancer. Okay, let's get into stem cells. Finally, phases of mitosis in animal and plant cells. All of us have what are known as stem cells. Many mammalian organs contain what are known as stem cells. Basically, stem cells, you guys, are like master cells. These are master cells. They retain the ability to divide. Red bone marrow stem cells divide to produce various different types of blood cells. Okay. We have therapeutic cloning and reproductive cloning. Therapeutic cloning is to produce human tissues that can begin with either adult stem cells or embryonic stem cells. Embryonic stem cells can be used for reproductive cloning or the production of a new individual. Scientists right now are looking a lot into therapeutic cloning. And when we talk about therapeutic cloning, it's basically using our own cells to harvest our own types of tissues. If we can harvest our own tissue, then we can repair damage to organs. All right, same thing. Here's another video on what stem cells are. So watch this, please. All right, the difference between reproductive and therapeutic cloning. So let's take a look. In reproductive cloning, the cells from the animal to be cloned are removed. As we could see here, the DNA is removed. The egg cell is now fused with the new nucleus. So we take this nucleus out and we add in a new nucleus, as we see here. Then we cultivate and we get these embryonic stem cells. We implant the embryo into a surrogate mother, and then the clone is born. Okay. In therapeutic stem cells, same thing. We get an egg cell. We remove the nucleus, right? And in this instance, we fuse the egg cell with the G0 nucleus. We let these embryonic stem cells grow, and these stem cells can divide into nervous cells, blood cells, and muscle cells. This one here is a clone of a full individual. In reproductive cloning. In therapeutic cloning, it's used for treatments to make new muscle cells, blood cells, nervous cells. How do cells become specialized? Here's another good video. It's going to talk about what are the signals that indicate which cells become what. How does a bone cell know to become a bone cell? How does it know to become a skin cell? Okay. All right, and that is it in terms of cellular division, regulation. I'm hoping you guys all learned something new. I know that this lecture was long, but again, hopefully you guys are pausing and going back and reviewing. All right, have a great day.